Throughout human history, few infectious diseases have left as profound a mark on societies and global health as influenza. Far more than the seasonal flu that circulates each year, influenza pandemics represent seismic events, periods when a novel strain of the influenza virus emerges, spreads rapidly across continents, and causes widespread illness and death. These pandemics are not merely medical phenomena. They are social, economic, and political crises that test the resilience of nations and the ingenuity of science. From the devastation wrought by the 1918 Spanish flu to the challenges posed by the 2009 H1N1 pandemic, influenza pandemics have shaped public health systems, spurred innovations in medicine, and left indelible scars on the collective memory of humanity. The story of influenza pandemics begins long before the modern era, with historical accounts suggesting that outbreaks resembling influenza occurred in ancient Greece and Rome. However, it was not until the late 19th and early 20th centuries that the world began to recognize and document pandemics with greater accuracy. The first well-documented influenza pandemic of the modern age occurred in 1889 to 1890, often referred to as the Russian flu. Originating in Central Asia, or Russia, this pandemic spread rapidly along newly established railway lines and steamship routes, highlighting how technological progress could inadvertently facilitate the global transmission of disease. Over the course of several months, the Russian flu swept across Europe, North America, and beyond, causing an estimated 1 million deaths worldwide. This pandemic set a precedent for the speed and scale at which influenza could travel in an increasingly interconnected world. Yet it was the 1918-1919 influenza pandemic, the so-called Spanish flu, that truly revealed the catastrophic potential of this virus. Emerging in the final months of World War I, the Spanish flu was caused by an H1N1 influenza A virus of avian origin. Unlike most influenza outbreaks, which tend to disproportionately affect the very young and the elderly, the 1918 pandemic had an unusual mortality curve, with high death rates among healthy adults aged 20 to 40. This peculiarity remains a subject of scientific inquiry, with hypotheses suggesting that the robust immune response of young adults may have contributed to fatal complications, such as cytokine storms. The impact of the Spanish flu was staggering. In the span of little more than a year, an estimated 500 million people, one-third of the world's population at the time, were infected. Death toll estimates range from 50 million to as high as 100 million, making it one of the deadliest events in human history. The pandemic's effects were felt in every corner of the globe, from densely populated cities to remote villages. Societies were upended as hospitals overflowed, essential services collapsed, and entire communities were decimated. The war-weary world struggled to cope, and the limitations of contemporary medicine became painfully clear. There were no vaccines, no antiviral drugs, and only rudimentary understanding of viral transmission. Public health responses ranged from mask mandates and quarantines to the closure of schools and public gatherings, but these measures were implemented inconsistently and often too late to stem the tide. The aftermath of the 1918 pandemic left deep scars and important lessons. It underscored the need for robust public health infrastructure, international cooperation, and scientific research into the nature of influenza viruses. In the decades that followed, scientists made significant progress in understanding influenza, isolating the virus in 1933 and developing early vaccines during World War II. Yet the threat of pandemic influenza remained ever present as the virus continued to evolve and periodically jump from animals to humans. The next major influenza pandemic struck in 1957, known as the Asian flu. This pandemic was caused by an H2N2 influenza A virus that originated in East Asia, likely through reassortment between human and avian influenza viruses. The Asian flu spread rapidly across the globe, aided by increased air travel and post-war mobility. Although less deadly than the Spanish flu, it still resulted in an estimated 1 to 2 million deaths worldwide. Importantly, the Asian flu marked a turning point in pandemic response. For the first time, scientists were able to quickly identify the virus and develop a targeted vaccine within months. This achievement reflected advances in virology, surveillance, and international collaboration, laying the groundwork for future pandemic preparedness. Just over a decade later, the world faced another pandemic, the Hong Kong flu of 1968 to 1969. Caused by an H3N2 influenza A virus. Like its predecessor, the Hong Kong flu emerged through genetic reassortment, combining genes from avian and human influenza strains. The pandemic spread swiftly, facilitated by global travel and urbanization. While its mortality was lower than previous pandemics, estimated at around 1 million deaths, it demonstrated the persistent threat posed by influenza's capacity for genetic change. The 1968 pandemic also highlighted improvements in public health response, with rapid vaccine development and distribution helping to mitigate the impact in many countries. Despite these advances, influenza pandemics remained unpredictable and difficult to control. The emergence of new influenza subtypes in animals, particularly birds and pigs, continued to pose a risk of future pandemics. In the decades following the Hong Kong flu, scientists and public health officials intensified efforts to monitor influenza viruses in both human and animal populations, develop stockpiles of antiviral drugs, and improve vaccine manufacturing capacity. These preparations would be tested in the 21st century with the arrival of the next pandemic. In 2009, the world confronted the first influenza pandemic of the new millennium, the H1N1 swine flu pandemic. Unlike previous pandemics, which had emerged from Asia, the 2009 H1N1 virus originated in North America, arising from a complex reassortment of swine, avian, and human influenza genes. The pandemic was characterized by rapid global spread, with the World Health Organization declaring a pandemic just weeks after the initial cases were identified in Mexico and the United States. The 2009 pandemic was notable for its relatively mild severity compared to earlier pandemics, with estimated deaths ranging from 150,000 to 575,000 worldwide. However, it disproportionately affected younger age groups and pregnant women, and it placed significant strain on healthcare systems. The response to the 2009 pandemic reflected both the strengths and limitations of modern public health. On the positive side, global surveillance networks detected the new virus quickly, and vaccine production began within months. Many countries implemented pandemic preparedness plans, distributed antivirals, and launched public information campaigns. However, challenges persisted. Vaccine supply was initially limited, and there were disparities in access between high-income and low-income countries. The pandemic also exposed gaps in communication, coordination, and the ability to adapt to rapidly evolving situations. Despite these challenges, the 2009 H1N1 pandemic ultimately caused far fewer deaths than initially feared, and it provided valuable lessons for future pandemic planning. Beyond the major pandemics of the past century, influenza has continued to pose a constant threat through seasonal epidemics and the emergence of novel strains with pandemic potential. The highly pathogenic avian influenza H5N1, first detected in humans in 1997, has caused sporadic outbreaks with high mortality rates, though it has not yet achieved sustained human-to-human transmission. Similarly, other avian influenza viruses such as H7N9 have caused concern among public health officials. The ongoing risk of zoonotic spillover, whereby influenza viruses jump from animals to humans, underscores the importance of surveillance at the human-animal interface and the need for a One Health approach that integrates human, animal, and environmental health. The persistence of influenza pandemics is rooted in the biology of the virus itself. Influenza viruses are RNA viruses with segmented genomes, allowing for frequent genetic reassortment, a process whereby gene segments from different viruses mix to create new combinations. This ability to shuffle genetic material enables influenza to evade immunity from previous infections or vaccinations, leading to the periodic emergence of novel strains capable of causing pandemics. Moreover, the natural reservoirs of influenza viruses in wild birds and the close contact between humans, poultry, and swine in many parts of the world create opportunities for cross-species transmission and genetic mixing. The societal impact of influenza pandemics extends far beyond the immediate toll of illness and death. Pandemics disrupt economies, strain healthcare systems, and challenge the social fabric. During the 1918 pandemic, for example, many cities experienced labor shortages, school closures, and interruptions to basic services. Public gatherings were banned, businesses shuttered, and funerals conducted hurriedly or in mass graves. The psychological toll was immense, with fear, grief, and uncertainty permeating daily life. In subsequent pandemics, the economic costs have also been significant, with lost productivity, increased healthcare expenditures, and disruptions to trade and travel. Public health responses to influenza pandemics have evolved considerably over the past century. Early responses relied heavily on non-pharmaceutical interventions, such as quarantine, isolation, and the use of face masks. While these measures remain important, advances in virology, immunology, and biotechnology have enabled the development of vaccines and antiviral drugs as key tools in pandemic control. Seasonal influenza vaccines are now produced annually, tailored to the circulating strains predicted through global surveillance. During pandemics, the rapid development and deployment of vaccines are critical to reducing morbidity and mortality. However, challenges remain in ensuring timely production, equitable distribution, and public acceptance of vaccines. The experience of recent decades has highlighted the importance of global cooperation in pandemic preparedness and response. The World Health Organization, national public health agencies, and international networks such as the Global Influenza Surveillance and Response System, GISRS, play vital roles in monitoring influenza activity, sharing data, and coordinating responses. The establishment of the Pandemic Influenza Preparedness Framework has sought to ensure fair access to vaccines and antivirals, particularly for low and middle-income countries. Nevertheless, disparities in resources, infrastructure, and political will continue to pose obstacles to effective global action. The COVID-19 pandemic, caused by a novel coronavirus rather than influenza, has further underscored the challenges and imperatives of pandemic preparedness. Many of the strategies employed, such as social distancing, mask wearing, and the rapid development of vaccines, echo those developed in response to influenza pandemics. The experience of COVID-19 has renewed interest in universal influenza vaccines, which aim to provide broader and longer-lasting protection against diverse strains. Research into mRNA vaccine technology, accelerated by the COVID-19 response, holds promise for improving the speed and efficacy of influenza vaccine development in the future. Looking ahead, the threat of influenza pandemics remains real and ever-present. Changes in global demographics, urbanization, climate change, and increased movement of people and animals all contribute to the risk of new pandemic strains emerging. The lessons of past pandemics, both their tragedies and triumphs, offer guidance for the future. Investment in public health infrastructure, research, and international collaboration is essential. Strengthening surveillance, expanding vaccine manufacturing capacity, and ensuring equitable access to medical countermeasures are critical priorities. Education and communication also play vital roles in pandemic preparedness. Public trust in health authorities, willingness to adopt preventive measures, and acceptance of vaccines are shaped by effective communication and transparency. Misinformation and vaccine hesitancy can undermine response efforts, as seen in both influenza and COVID-19 pandemics. Building resilient communities requires not only scientific innovation, but also social cohesion and trust. In conclusion, the history of influenza pandemics is a testament to both the power and vulnerability of human societies. Each pandemic has brought suffering and loss, but also spurred advances in science, medicine, and public health. The interplay between viral evolution, human behavior, and societal response shapes the course of each pandemic, and the lessons learned inform our preparedness for future threats. As we look to the future, the challenge is to harness the knowledge, tools, and solidarity needed to confront the next influenza pandemic, whenever and wherever it may arise. The story of influenza pandemics is far from over, but by learning from the past and investing in the future, we can strive to write a new chapter marked by resilience, innovation, and hope.